Hello, my name is Ross Cummings, and I am a UK and European patent attorney at Gill James and Every, a leading firm of European patent attorneys in the UK and Munich. This is part two of this expert talk on gene therapy. In part one, I took you through some of the history of gene therapy, and I gave some financial context to the current cell and gene therapy market. I'll now discuss some of the challenges for patenting in this field in Europe, and I will set out strategies for addressing these. There are a number of hurdles in the European patent system that must be overcome to get biotechnology patents granted. Some of these are accentuated with gene therapies, but it's still essential to protect your innovations, and there are ways around the problems. Patents are often essential for startup biotech companies to support fundraising for their fledgling technologies. The companies may have started at a university where early funding was available, potentially to get to preclinical proof of concept. But usually when it comes to raising much larger sums of money required to start scaling up, developing the technology, and moving into phase 1 clinical trials, external investment is required. From experience, having defensible patents is key to surviving due diligence. This might seem challenging in gene therapy, as gene therapies involve manipulating and using parts of the human body which are not patentable in Europe. And gene therapy also involves methods carried out on the human body which are also not patentable in Europe. However, these are not actually significant hurdles to patenting these technologies in Europe. Perhaps a greater problem is that the technology has not moved on significantly from when it was first developed over 30 years ago. The patent cannot grant unless the invention is new, so the exact details of what you are developing become very important. According to the patent office, biological processes are inherently unpredictable. This makes extrapolation of experimental data to broad patent claims challenging. This is accentuated with gene therapy due to the various problems that have been encountered in clinical trials to date. You may be able to use your vector to drive stable expression of your transgene in the lab, but do you know that it would work in the clinic? And recent problems with the efficacy of Roctavian demonstrate this. Therefore, how do you show in the patent application that the invention actually works? This is a key requirement for a granted patent. If you can show that the invention works, can you show that it would still work if a slight change were made to it? Do you have data to support this? If not, this may mean your patent claims need to be quite narrow. Is this a problem? Well, the answer is that it depends. In a field beset with scale-up challenges, if you solve these problems and get process patents granted, will your patents be of value and will you be able to enforce it? The prohibitions on patenting methods of treatment and parts of the human body are the easiest issues to address. While you cannot patent methods of treatment, processes for modifying the germ-line genetic identity of humans or use of human embryos, with gene therapies, you can avoid these exclusions by focusing your patent claims on the products that you manufacture in the lab. In other words, you can patent the nucleic acid constructs, vectors, and modified cells that are manufactured as part of the process to provide a gene therapy. However, unlike with other types of medicament, it is less likely that second medical use patents will be particularly important. In other words, using known products for different indications. This is because firstly, the products are specific to the disease, and secondly, once you know a vector is effective for gene therapy in a particular tissue, replacing the transgene to treat a different indication is likely to be considered obvious, although this will depend very much on the facts. Nevertheless, this may mean a slightly different patent strategy is needed to compare with other medicaments, particularly for broad platform patents, but this will come down to the facts again. You can patent isolated genes, but usually only if you have modified them in some way or included them within a larger construct. With ex vivo gene therapy, you could obtain a patent to a particular type of cell, for example, a liver cell, overexpressing your transgene. This would depend on the fact. And biological deposits are not likely to be useful for patenting purposes here, as the actual therapy is based on the patient's own cells and not generic cell lines. Patents to the vectors are clearer and broader in any event. Lastly, any novel reagents and processes devised in your development could be patentable. Indeed, these types of patents could be very useful, as they would likely apply to not only your own technologies, but also the technologies of competing organizations. This could represent additional funding streams through licensing royalties if you choose to go down this route. Also, both of the currently approved gene therapies use an adeno-associated virus, and most of the gene therapies in the pipeline do too. This technology is not that dissimilar to the technology that was developed over 30 years ago. There are 9 serotypes of adeno-associated virus, and they differ in their affinity for different tissues in the body. So this is a factor in selecting the right one for your therapy. It's actually quite unlikely that a biotech company developing a gene therapy will have actually discovered the genetic cause of the condition that they're seeking to treat. Such basic research is usually carried out in an academic setting and published. Therefore, unless you have improved the vector more generally, broad patent protection for treating a particular disease by restoring the function of a particular gene will probably not be available. But many patents will claim the actual vector plus transgene product quite narrowly. This does not diminish the value of these patents, though. The tight regulatory regimes around these products mean that even if your patent is very narrow, as long as it covers your product, it will still offer investors comfort and hold off generic competition while it's in force. Indeed, a company seeking to manufacture a gene therapy with a completely novel vector is likely to face much longer development times and more regulatory scrutiny, as they will not be able to benefit from the acceptance of vectors already in use or in clinical trials. Therefore, there are not only technical hurdles to innovating in this area, but also regulatory ones. Many larger corporates are looking into novel vector technologies, but it seems likely that it will be a long while before we see this in the clinic. Harvard University spin out Dino Therapeutics has generated a lot of interest recently. They are looking at improvements in vector design and recently raised $222 million in an IPO. Now, there is an invisible yet crucial plausibility line that must be crossed in Europe before you file a patent application. This line is there with all technologies, but with most it's clear and present on formation of the inventive concept. It's much more subtle in biotechnology and pharma, yet increasingly more critical for getting and keeping a granted patent. Sufficiency of disclosure is the requirement that a patent application provide a skilled person with sufficient information to enable them to reproduce the claimed invention. For the patent application to be sufficient, it must be plausible, based on the contents of the patent application, that the claimed invention would work. This is often a matter of claim breadth when compared with the experimental data in the application. And does the experimental data in the application support the breadth of claim? If you overreach, you may not be sufficient. So you need to make sure that the patent application is drafted with this in mind. This issue was recently highlighted to the detriment of the patent owner in Regeneron versus Kymab in the UK Supreme Court. When drafting your claims, you have to consider whether, with the technology available at the moment, it is possible to perform the invention across the claim scope. Data can take time to put together, and the ultimate data in gene therapy is the result of a clinical trial. However, you can't wait that long to file your patent application at the patent office. Simply put, if you file your patent application too early, your invention may be insufficient and never get granted, or if you manage to get it granted, vulnerable to revocation. However, as is the case with any competitive and rapidly developing art, if you file your patent application too late, then you may be beaten to it by your competitors, or make an inadvertent public disclosure and your patent application will fall on lack of novelty or inventive step. Indeed, if you wait for clinical trial data, then you will inevitably be destroying your own possibility of getting a granted patent, as a clinical trial process involves non-confidential disclosures usually. In gene therapy, a construct may perform well in the lab but fail in clinical trials. There are many reasons for this, such as a difficulty in getting the right dose to the right place, the efficacy of the vector in the target tissue, the longevity of expression, host immune responses to the vector, or the transgene product indeed, and many others. You cannot predict these clinical outcomes from preclinical experiments. However, it's important to note that a risk of failure is different to a lack of plausibility. These are two separate matters under European patent law. Sufficiency of disclosure is an assessment of whether it is plausible that it would work based on the experiments in the patent application. Whether it actually works is instead a matter of inventive step. Now, inventive step is the requirement that a claimed invention is not obvious over what has gone before. Curiously, Unlike sufficiency and plausibility, you can provide your evidence that your invention actually works and is not obvious later on down the line, crucially, after filing your patent application. Therefore, there is a level of data required for sufficiency of disclosure, and then a second level of data required for inventive step. The key is recognizing when you have hit the first, and then ensuring that later down the line you hit the second. There are strategies for delaying examination of your application in the event that the data is slow to emerge, but you can't do this indefinitely and it can become very expensive. And the content of the patent application is crucial, not only in terms of experimental data, but also in terms of the way in which the invention is framed. The application has to be flexible enough to be able to adapt to future events to allow you to arrive at an allowable claim. I've been working in the patent profession for over 15 years now, and over that time, I've noticed that the EPO has been examining biotech inventions in a steadily stricter fashion. The bar for supporting your claims with a specific experimental data has steadily increased to the point where there are definitely a lot of granted patents out there that would not have been granted under the current regime. The assumed inherent unpredictability of the effect of therapeutic biologics means that now, more than ever, in order to achieve grant in Europe, you need to narrow your claims to the specific product being trialed. This usually comes down to specifying nucleic acid or polypeptide sequences of the product in your claim. It used to be possible to generalize your invention in the fashion that my chemistry colleagues are accustomed, but not anymore, sadly. For example, if you are claiming novel antibodies or T-cell receptors, you have to specify the sequences of all the complementarity determining sequences in your main claim. The same applies to new cell and gene therapies. With a new cell, you might have to go as narrow as a biological deposit, but this depends on the specific technology in question. With a new gene therapy construct, you will likely have to specify the actual nucleic acid sequence, including the transgene, promoter, and regulatory sequences in your claim, unless you have generated data justifying broader claims. Again, this will depend on your actual scientific contribution. In any event, it's all about data. If you have appropriate data, you can still get what we call functional claims, where the molecule in the claim is defined by its activity rather than its structure. This is the holy grail of biotech patents, and in practice, very hard to justify without specific data and a strong scientific rationale. In the absence of any data in the application, then it's unlikely that mechanistic arguments alone, however good, will suffice to show plausibility, let alone inventive step. Therefore, if you file a patent application to a biologic medicine that contains no experimental data at all, it may never achieve grant in Europe. Now, there is an often used cliche in gene therapy. The process is the product. This is a nod to the complexity inherent in the chemistry and manufacturing controls and the exacting standards necessary. The problem itself is also more akin to an engineering problem, contrasted with the traditional pharmaceutical approach, where a screening process leads to the discovery that a random compound has efficacy in an assay and may be useful in treating a disease, allowing you to claim broadly around this compound on the supposition that similar compounds may have similar effects. In gene therapy, the contribution to medicine may not be the compound as such, but the process for making that compound and successfully demonstrating efficacy in treating a disease. The inventor starts with the knowledge that the target disease is caused by a particular mutation. And the problem is, how do we correct this in the clinic? In some instances, it may be the behind-the-scene processes, reagents, and tools that may form the basis for effective patent protection, rather than the construct itself. Of course, there are many instances where the invention is actually a novel aspect of the construct, so the strategy will depend on the facts. For example, the recent Bristol University spin-out PureSpring Therapeutics has received £45 million in funding to further develop their kidney-targeted gene therapy. Their technology is based in part on particular promoters that can be used to drive expression of their transgene in kidney tissue. Now, there are many problems to be solved in gene therapy, and therefore many opportunities for effective patent protection. Key problems include improving transfection efficiency in particular tissues of interest, improving maintenance of the vector and host cell, reducing off-target expression and toxicity, reducing immune reactions, addressing production cost issues, and of course, showing therapeutic efficacy over time. Process patents will be important in some of these instances, particularly in addressing manufacturing challenges. Solutions to this problem would be relevant to many different products, so it could be very useful IP. Inventors developing chemistry, manufacturing, and control improvements for gene therapy may not actually realize that they are inventing patentable technologies while solving manufacturing problems. So it's important to provide education to your scientists to ensure that these inventions are captured in patent applications before they are disclosed in journal articles or regulatory submissions. Now, one advantage of having patents covering these behind-the-scene aspects is that they may be product agnostic, so may actually apply to multiple products or potential products, as well as your competitors' products. Such IP may provide strong licensing revenue while you're still in the development stage of your pipeline, as other companies may want to benefit from your discoveries for their own potentially non-competing products. This is worth considering in your commercial strategy in any event. Now, in part 3 of this expert talk, I'll take you through some particular case studies that illustrate some of the issues I've been discussing. Thank you very much for listening.